to the environmental engineering seminar. And Dr. Seegren asked me to announce at the beginning that if you want continuing education credits, just email him and tell him which seminar you attended, and he can send you an official certificate so you can use it for your continuing education. Today we have the honor to welcome Dr. Renee Richer. She received her BA in biology from the University of Chicago and her Ph.D. in biology from Harvard University in 2004. She joined the University of Wisconsin Green Bay Marinette campus in August 2014. Her current work focuses on the intersection of biological processes, sustainable development, and human health. For the last decade, she's been working to understand routes of human exposure to cyanobacterial neurotoxins, such as those produced by harmful algal blooms, and their role in ALS, Alzheimer's and Parkinson's disease. So with that introduction, I'll turn it over to you, Renee. Go ahead.
Great, thank you. And thank you so much for having me. So as you can see from the title, from Qatar to Calumet, I'm trying to draw the link between a lot of the work that I did in the Middle East directly and the link between water quality issues here in the area, specifically with the Great Lakes. So I spent approximately 8 years working in the Middle East in Qatar, and it's a hyper-arid environment. And so your first inclination might be to question the relationship between environmental issues in these two very different environments. But by the end of today, hopefully you'll see the connection. All right, so this story, the relationship between cyanobacterial neurotoxins and neurodegeneration actually begins in Guam in the late 1940s. So you can see Guam pointed out there with the red arrow. And this is a very small island in the Pacific. And after World War II, doctors were in the area, and they were seeing that as much as 25% of the population was actually dying from this neurodegenerative disease that had very much the features of ALS disease, Alzheimer's, and Parkinson's. And so there was a lot of question about, what was causing these very high rates of neurodegeneration. And work done since there has shown that it was tied to their local diet. And it turns out, on the bottom here of the screen, on the very bottom, you can see roots of a local cycad plant. And cyanobacteria, those are photosynthetic bacteria, also known as blue-green algae. And they form a symbiotic relationship with the plant. But cyanobacteria also produce really potent toxins, and some of those are really potent neurotoxins. And that toxin would be incorporated into the seeds of the cycad. And the local diet was actually based on a flower, which was produced from these seeds. Also in the upper right-hand picture, you see a flying fox there. They would also consume these seeds. And the flying foxes would incorporate the toxin into their bodies. Now, the toxin that I'll be talking most about today is L-BMAA, which is beta-methylaminoalanine, and that's a modified amino acid. So this is what really started the research into this field. And for many years, we simply didn't have the analytical tools to understand how this neurotoxin was acting and how it was infecting the population. All right, so I want to bring you to Qatar. And Qatar is this small peninsula off of Saudi Arabia, off the Arabian Plate. And it's a hyper-arid desert. The northern part of the country receives about 100 millimeters a year in rain. The southern part receives about 40 millimeters of rain. And you can see what the landscape looks like there. So it is a very dramatic and very extreme type of landscape. And when I first arrived in Qatar, my specialty was looking at plants. I was largely a plant ecologist who had done work on issues of climate change and other environmental issues. But when I arrived in Qatar, it turns out that the dominant photosynthetic organism was in fact Cyanobacteria. So let me just go on. What are Cyanobacteria? Cyan means blue-green, and these are photosynthetic bacteria, some of the first known organisms on planet Earth, responsible for producing much of the oxygen in the atmosphere. Previously, they would have been known as blue-green algae, but now we, in fact, recognize that this is a photosynthetic bacteria. So in the landscape here, in the landscape image, you see lots of what looks like dry, cracked mud, but that's actually this complex microbial community comprised largely of cyanobacteria. And up in the upper right-hand corner, you see oxygen evolution. So they perform oxygen evolving photosynthesis like our plants. And they can be filamentous, like you see in the lower right-hand corner there. But they really dominate the landscape. And much of the satellite imagery we get from desert areas, when we see that great greening of the desert during times of rainfall, that's actually primarily a signal from cyanobacteria rather than from plants. This is an image from Carrara, which is an area in the southwestern part of the country. And this is particularly interesting because the quartz rocks allow just enough light to pass through for the cyanobacterial communities to survive on the bottom portion of the quartz rocks. And again, you can see we have single cell and filamentous cyanobacteria there. And this has only been documented in one other place in the Namib Desert. And so this is quite a unique community of cyanobacteria. And this is the inland sea region. So this is an area where you have very, very shallow sea in the southeastern part of the country. And as far as you can see, there are these thick, black cyanobacterial mats. So the country itself is really dominated by cyanobacteria. So I was working with a number of different colleagues, and me being in Qatar, they were starting to pursue in earnest this idea that exposure to cyanobacterial neurotoxins could be linked with ALS in particular, and ultimately Alzheimer's and Parkinson's. So this was the first work that we published in 2009 looking at Gulf War syndrome. And what we proposed at that time was that the one single biological agent that could be tied to Gulf War syndrome in vets from the first Gulf War was in fact exposure to cyanobacteria. And so you can see here, cyanobacteria and BMAA exposure, a possible link to sporadic ALS. So in Gulf War veterans, we see twice the rate of ALS, and that usually has a time-limited outbreak. So within 10 years of their exposure, we see increased rates of ALS only in those that were deployed to the Middle East. And so this was really the first work that we published from the area. And can I say, if anybody has a question, you can keep them to the end, or if you want to jump in and ask a question at the time, I'm open to that as well.
I have a quick question,
Sure.
Renee. Did they see anything similar during World War II with all the fighting in North Africa? Were there any indications of similar?
Not that I'm aware of, no. But in North Africa, depending on the environment, if it was more sandy, for example, you tend to see less cyanobacterial mats in that situation. Also, the question I would ask is, did they have adequate epidemiological follow through? In that case, I'm not quite sure, but I'm not aware of that link.
Yeah, thank you.
And And in fact, if I comment on that, if I remember correctly, they did see increases in rate of ALS, but it only became significant when soldiers are deployed to the Middle East. All right, so I started to ask some simple questions. I was working with a number of colleagues, particularly Paul Cox and James Metcalf, Aspa Hatsiepmu, Sandra Bannock, etc. And I asked a simple question: where is much of the cyanobacteria located in Qatar? So we did a nationwide survey. We covered more than 9,000 square kilometers, covering, I think, nearly 90% of the land surface area of the country. And what we saw was that in the north, where we have greater rainfall and there are topographical depressions, in the landscape, we have much higher levels of cyanobacterial coverage. So as much as nearly 90% of the land surface area is covered by this cyanobacterial mat. And as you move further south and it becomes drier, you get a lot more in regards to sand dunes. And so the substrate is shifting and you see fewer cyanobacterial mats. So we have a strong grain gradient as well as topographical gradient in regards to where the cyanobacterial mats are. But the important point here is that, you know, there's the potential for the population to be exposed to cyanobacterial toxins quite easily. And then we asked, well, what toxins are actually present in the environment? So we know that there's a lot of cyanobacteria. It covers vast areas of the desert. What might people be exposed to? And so we identified a number of toxins from those samples. Microcystin LR, if any of you are familiar. This is a liver toxin. It's also a tumor promoter. And this is probably our best known cyanobacterial toxin. This is what shut down the water supply to Toledo when we had the harmful algal bloom in Lake Erie. And again, this is what we're more accustomed to thinking of when we think of cyanobacterial toxins. We also identified anatoxin AS. And this is an acute neurotoxin. It's also known as very fast death factor. And it acts very much like sarin gas as our chemical weapon, sarin gas. And so you see a very quick onset of dramatic neurotoxin poisoning from exposure to anatoxin AS. The next three neurotoxins include BMAA, that's beta-methylaminoalanine, and two isomers, diaminobutyric acid and amino ethyl glycine. And these are modified amino acids. And so we still have a lot of work to do to understand the pathways by which these amino acids are produced and the environmental conditions under which they're produced. But these are what we identified. So BMAA, DAB, AEG, those are all neurotoxins, but slower acting neurotoxins that can accumulate, and anatoxin AS. So then we asked, well, what is the exposure risk? So if you're living in an environment which is dominated by cyanobacteria, and they produce these very potent toxins, what is the risk of being exposed to these toxins? And the first question we asked is through air. And if you are familiar with the Middle East at all, you might be familiar with sandstorms, which you see here illustrated in this image. And I can assure you that it's very much like this. It is a wall that comes and engulfs you. And sometimes you may be in the middle of a sandstorm for 5 days, even more. And so we did some basic calculations looking at the levels of microcystin in this case in the soil and how many times your average person breathes per minute, and came up with some calculations and found that in a single 24-hour period, your average adult would be exposed to a very significant level of microcystins. If it was a young child, they would exceed the tolerable daily intake for exposure to these toxins. So even through air, you can have a significant exposure. And I'll come back to that again when we talk about lakes. So then we ask the question, well, where are these toxins found? And the first place we looked, obviously, was in the soil. So the cyanobacterial mat, it's almost like leather. It has the consistency of leather, and it's this complex microbial community. So where exactly do we find those toxins? And what we see in this image here on the left is that our 3 neurotoxins are actually found in the soil column, up to 25 centimeters. And so these toxins accumulate in the environment. They're not easily broken down because they are amino acids. And so exposure can come from exposure to the soil. So particularly those that are working in construction. If the soil is aerosolized, there's the potential for exposure there. And you can see the water table. And we then asked, could we potentially have contamination of groundwaters and other waters from these toxins, which are held at depth in the soil. So we went throughout the country and we looked at water supplies. Now, the country in Qatar is growing so fast. It has one of the highest population increases in the world. And a lot of the infrastructure, basic infrastructure, isn't keeping up with that growth of the country. So water tends to be stored in containers, both in the urban environment as well as in the rural environment. And so we did samples, extensive sampling of water storage containers in the rural environment as well as in the urban. And what we found in the urban environment is that most of the water storage containers are made of fiberglass, and they tended to contain microcystine, and microcystine in levels that exceeds the World Health Organization's tolerable daily intake levels. So we tend to see quite a bit of microcystine in those storage tanks. When we went out to the rural areas and sampled storage tanks, what we found was that, in fact, we were identifying AEG, which is one of our slower acting neurotoxins, amino ethyl glycine. So again, within the water supply, we're finding these types of cyanobacterial toxins. All right, so working in the country, working in the field, we spent a tremendous amount of time, my colleagues and I, and here in the image you see my colleague, Dr. Aspa Hatziefdemiu, and that's my daughter, and that's Aspa's lovely dog named Star. And we were out doing field work in the field, and there was a cyanobacterial mat, and we had recently had rain. So there was a small rain pool sitting above the soil on this cyanobacterial mat, and Star, the dog, drank a little bit of that water and immediately started to show acute signs of neurotoxin poisoning. So we put the dog into the vehicle, rushed to the veterinarian. And I must say it was the fastest I've ever driven in my life. We managed to get to the vet and the dog survived. We tried to convince the vet that in fact the dog was suffering from neurotoxin poisoning. She did not believe us. She thought I was crazy, not realizing in fact that's what we were studying. But it turns out that a number of dogs had died that season. And later, after everyone had calmed down a little bit, we came back to the vet and we ultimately published this paper, which was the first known exposure of an animal to cyanobacterial toxins in desert environments. So typically we think of cyanobacterial toxins as associated with water bodies. And so what we're seeing is that, in fact, you can have acute types of poisonings or exposure to these toxins in desert environments as well. So the fact that a mid-sized dog could be poisoned so dramatically, we began to express concern about children that may be out in the desert playing in these small rain pools and what they might potentially be exposed to. So continuing to ask the question, where are these cyanobacterial toxins found? We began to look in the food chain. And again, we're more familiar with cyanobacterial toxins and exposure in aquatic or marine environments. So we did a comprehensive survey of the key marine species that were used for human consumption, some of the most favored food species. And we were looking for biomagnification. And what we didn't find is at the base of the food chain, we didn't see a lot of toxin accumulation. But as we move further up the food chain, we saw diaminobutyric acid and amino ethyl glycine accumulating in some of the fish species. So another potential route of exposure is through marine organisms or aquatic organisms. And one question that we asked is if you have, let's say, disturbance of the environment. If you disrupt that thick crust that covers the desert soil, does that greatly increase your risk of exposure? And what we found here was that, in fact, if you have a pristine environment where the cyanobacterial mat itself is not disturbed, that you greatly reduce bioaerosols in a wind event. So again, if we can maintain that surface of the desert soil, maintain that soil crust, it greatly reduces exposure to bioaerosols. This is unpublished data here. We also asked the question, well, does traditional dress potentially protect people from exposure to cyanobacterial and the toxins? And again, this is unpublished data here, but we did find that there was some increase in exposure to antitoxin and also particularly chlorophyll A, which we use as a measure of cyanobacterial, the presence of cyanobacteria or density of cyanobacteria. And so we did see that there was some protective impact. And this is something that, you know, we should in fact pursue more strongly, that traditional dress seems to protect the population at least somewhat from exposure. So in Qatar, what we found was that these very potent neurotoxins were present in the soil and to depth in the soil, in the water that was being consumed, both in rural and urban environments. It was present in the food that was being consumed, marine or aquatic organisms, as well as in the air that we breathe. So the question then is, what is the link with Michigan waters, right? So here we are in Michigan, we're surrounded by the Great Lakes, and what is the link? And of course that link is harmful algal blooms. So we know that the duration, frequency, intensity of harmful algal blooms is increasing on the Great Lakes, which I'll talk about in a little bit more detail. But the organisms, here's the important point, the organisms that are the key component of harmful algal blooms are the same cyanobacteria that me and my colleagues were studying in the Middle East. So all of those toxins and potential routes of exposure translates very nicely to work done here in Michigan with harmful algal blooms. So these are just some images here showing the Green Bay. You can see Cyanobacteria or harmful algal bloom on Lake Erie on the top left, and it looks very much like pea soup. So the water has the appearance, the consistency of pea soup, and that is when you get a bloom or a great growth of this photosynthetic bacteria. Now, right now, the Green Bay is one of 3 freshwater areas being monitored by NASA from space to predict the potential outbreak of algal blooms. And so it's playing quite an important role in that regard. So this is work that was done by colleagues, and they did some animal model work, and they fed animals BMAA, beta-methylaminoalanine. The Cyanobacteria produce neurotoxin, which I've been talking about. And what they found is that exposure, dietary exposure to this neurotoxin produces neurofibrillary tangles and amyloid plaques. And in fact, it's the first animal model to show that. So typically it's humans that show these key features of neurodegeneration. And this was the first animal model that shows that type of production of neurofibrillary tangles and amyloid plaques. What's also really important is that it was dose dependent. So depending on the amount of BMAA that the animal was exposed to, there was a greater concentration of neurofibrillary tangles and amyloid plaques in the brain material. And of course, these are the key features of ALS disease, Alzheimer's disease, and Parkinson's disease. And what's interesting in this paper as well, they were able to rescue some of the animals. And that rescue came from feeding the animals another amino acid, L-serine in particular. So I want to talk a little bit about that. Here's another paper. This isn't published by me. This came out relatively recently. And what it was looking at was the SOD1 protein. So this is superoxide dismutase, which is very important in general neuroprotection. And the vast majority of ALS cases are sporadic, meaning there's no known cause. Only about 5% of ALS cases are known to have a genetic cause, and that is usually linked with the superoxide dismutase and a genetic mutation with the SOD gene. And so in this modeling, they looked at the protein, the SOD1 protein, and if that amino acid, BMAA, was accidentally incorporated instead of the standard amino acid L-serine into the protein, you would get protein misfolding. So that's a very important aspect here, is that these toxins, they can bind to receptors, they can cause overexcitation of the neurons, which can lead to neuronal death, but also because it's a modified amino acid, what happens is it gets accidentally incorporated into the protein, and that can cause protein misfolding. So there are a number of different mechanisms by which these toxins can actually cause neuronal death and neurodegeneration. All right. So in regards to future considerations, we know that phosphorus in particular is what's driving harmful algal blooms. Also, warmer temperatures associated with climate change and increased rainfall. So for every degree increase in temperature, we get about 7% increase in rainfall. And that causes greater phosphorus runoff from the landscape into our Great Lakes, into surface water sources. So while we cannot directly address issues of climate change, such as warmer water temperatures or increasing rainfall, until we develop sort of a federal strategy or global strategy to deal with climate change. That leaves us largely to deal with phosphorus. And right now that's being heavily debated. And there's some pushback against phosphorus regulations. There's a possibility of groundwater contamination from surface water sources. So there's just beginning to, we're just beginning to see this being addressed in the literature. And this is an area that I have not published on yet, but something I've looked at is how these different toxins move through sediment and which ones are most likely to move through sediment and potentially contaminate groundwater sources. And again, right now, microcystine would really be the only cyanobacterial toxin that's being tested for in the event that we have a harmful algal bloom. So hopefully in the future, we'll have more testing for these very important neurotoxins. One thing to consider as the lake levels rise and as rainfall increases, we're seeing wastewater discharges into the lakes. And again, that is going to be a significant factor in fueling harmful algal blooms. So how we manage our wastewater is going to become an increasingly important issue, particularly with high water levels and increasing rainfall. All right, I just want to make let me go back, I want to make one last comment. So what we've seen in this area is that there's an increase in neurodegeneration associated with eating fish from the Great Lakes. We know that surround the area of Lake Erie, and this data also comes, for example, from the eastern part of the United States, data also from Europe, that living on the downwind side of a lake, which routinely has cyanobacterial blooms, increases your risk of ALS. We see ALS hotspots associated with living on or near lakes which have cyanobacterial blooms, and consumption of any type of marine or aquatic organism in those lakes. And so again, that's more than national data, but that's international data as well. So this is bound to become increasingly important as we move forward. So again, a lot of this work was done in concert with great colleagues of mine, Paul Cox, Sandra Vanek, James Metcalf, Aspa Hatziaftamu, and I had a great team of students at the UW Green Bay Marinette campus who have put in a tremendous amount of work working on these types of issues. So I would love to address any questions that you may have.
All right, well, this was a virtual round of applause for you. In virtual meetings, it's hard to hear the thundering applause that you would have gotten, but you would have gotten it. Take my word for it. So thank you very much. Yeah, we have plenty of time for questions or arguments or whatever we want. Any questions from you guys?
I have a question. Can you hear me?
Yes.
Yeah.
Okay. Yeah. Thank you so much for great talk. Very interesting. First of all, my group also has been studying the fate of the free amino acid in natural aquatic environment under the sunlight, photo irradiations. So we haven't really looked at the toxicity, but first of all, maybe I have two questions. So you talked about the BMAA or some isomers, PAG or AEG, those like amino acids.
Mm-hmm.
So what are the typical fate of those amino acids? Because are they present as a free amino acid or more combined form in, I don't know, maybe groundwater or like a desert or maybe surface water? I was just wondering, first of all.
Yeah, so that's a fantastic question, which is, how do we usually see these in the environment? Are they in the free form or are they in the bound form? And so, for example, in water, in the aquatic system, you normally find them in the free form. But once they get incorporated into an organism, we find them significantly in a protein bound form. And that's really where the big step forward came, is that, you know, if you did tests looking for free BMAA or free AEG, you wouldn't often find it in high concentration. But once you do the protein hydrolysis, then you see it. So every time our proteins are broken down, you can get repeated exposure to this modified amino acid. And again, amino acids are going to be highly valuable in the environment, right? So that's a question that still we need to understand is, you know, how quickly are they removed from the environment by other organisms? And what are some other potential types of exposure or routes of exposure? A colleague of mine, Tim Downing in South Africa, is doing fantastic work on that, looking at exposure, prenatal exposure. He's done some fantastic work on that.
Thank you. So sounds like based on what you're mentioning, this amino acid itself doesn't really induce any neurotoxin. Because if you look at the structure, it's just a very simple amino acid, aliphatic, and maybe amine indicates some kind of a toxicity, or maybe they couple with a protein, and then for the first time they start showing some neurotoxin or.
Yeah, so actually what it can do, and it can in particular mimic GABA. And so you get binding depending on the receptor, right? So it can bind and over-excite. So you can get excitotoxicity from exposure to these. And what's interesting is in Guam, they would wash the cycad flower many, many times. And they would throw the wash water out. And if the chickens would drink the wash water and show a reaction, they would know that the flour wasn't yet ready for consumption. Or if they would consume it, they would start to feel that sensation. It acts very much like MSG. So if you go to, some people have Chinese food headaches, right? It acts very much in the same manner. So you can have excitotoxicity, but it can also then, you can have a second mechanism, which is misfolded proteins. And that's the area which is being explored in much more detail now. So most of the work that I've done has been looking at the roots of human exposure. How would they potentially be exposed to these toxins?
Great. Last question is, you also mentioned we still need to understand the pathways. So again, by looking at those structure, to me, it looks really like some kind of a microbial activity product. I mean, the BMAA or some of the isomers. So where do they come from? Are they just, you know, if you look at the, say, microcystin LR, that's a huge molecule. And then, you know, that probably we chunk off, and then we produce a lot of different amino acid or some other functional group. But they also part of the some kind of, you know, photosynthesis or maybe microbial activity byproduct, or I was just wondering, where do they really come from?
Yeah, so this is in some ways still an open question, but right now it's looking as if it's a molecule that helps to regulate carbon-nitrogen ratios. And depending on the amount of nitrogen in the environment, the amount of phosphorus in the environment, you can then get induction of the production of these toxins. So, you know, they don't produce these toxins under all circumstances. So it looks like it has to do with the carbon-nitrogen ratios. What we don't know yet is whether it's a primary metabolite or a secondary metabolite. And that pathway is yet still to be pursued.
Thank you so much.
This is a follow-up on that question, actually, and you may have answered this in your talk, but were there certain, you know, of course, not all cyanobacteria produce toxins under all conditions. Were there certain environments, you know, let's just focus on the desert, you know, the soil crust, where they were, toxin production was really high, or certain environmental conditions, or was there any pattern there that you saw?
Yeah, so this was some of the work. We did a bit of this work, and so what we see is, again, if we manipulate the amount of nitrogen in the environment, we can induce the production of this. Now, most cyanobacteria, 96% of species that have been studied, produce these toxins. And so it seems like this is a fairly conserved metabolic pathway. In the desert, though, it depends. In different environments, we see different isomers that dominate. So in Qatar, we see a lot of AEG. My colleagues working in Australia see a lot of AEG. And so you can see a preponderance of a different isomer. And again, it seems to somehow regulate carbon to nitrogen ratios. And so it can be driven by the amount of nitrogen in the environment or carbon in the environment. So cyanobacteria generally like more alkaline conditions because of the presence of the carbonate.
Right, right. Okay, thanks.
Let's open it up for the students to ask any questions they have. Students, anything? Kenny, I see your hand is raised. Go ahead.
Yeah, hi, my name is Kenny. I know that there are a lot of deserts in the world, but are you planning on looking at any of the deserts in the southwestern United States that have growing populations and are also on the eastern side of the jet stream in many cases? So wind does tend to come across the deserts into those more populated areas.
Yeah, so we have some colleagues that are working in that environment. The thing is, let's say in southwestern US, Utah examples, the rainfall is much higher, about 300 millimeters per year. And so what we see is that those microbial mats, or what we call biocrusts in desert environments, usually have a greater proportion of fungi associated with them. And when you go to the hyper-arid environment, you get a preponderance of the cyanobacteria in the biological soil crust, that crust that covers the top. So there are colleagues that are working in the Western US, in the deserts of the US, and they're finding the same types of toxins, et cetera. But again, the crusts are very different depending on the proportion of fungi, and that's tied to rainfall.
Thank you.
Any other questions from the students? Okay, well, I had one. Oh, was there a student question? Go ahead. Okay, well, so if we wanted to upgrade our water treatment plant processes or wastewater treatment plant processes to remove cyanobacteria or any harmful aspects, say like the city of Toledo might be very interested in, what would we have to add to our present day water treatment plants, for example, to remove this? Any thoughts on that?
So sand filters right now seem to be fairly effective. If you have something which has a lot of organic material in it, a lot of the binding sites are filled. But in some of the work that I've seen, again, this is a relatively new field, and we're starting to see this come out now, is that sand filters seem to be fairly effective in filtering out a lot of those. I think one of the challenges comes with testing because it can be difficult to distinguish between the isomers, for example, of the modified amino acids. So right now you can send samples to a variety of labs to be tested for cyanobacterial toxins, but it's not a test that's easily done in a desktop setting. You know, it would be nice if we had something like a pregnancy test, right, where you can dip the stick and you get a result, but right now we just don't have that. You can use ELISA for microcystine, but it comes down, right now, we just don't have very easily accessible testing for the neurotoxins. I know Wisconsin has put a lot into trying to disrupt harmful algal blooms. They've tried ultrasonic disruption, but what happens then is that the toxins get spilled out of the cells into the environment, and you get this large flush of toxins in the water. So, you know, disrupting the cells, breaking the cells doesn't seem to be yet a good way to remove toxins.
What about the granular activated carbon type of a system? Is that effective, do you know? Or?
Yeah, I think it would be, but I know less about that in regards to removing from water.
Yeah. Well, also, if we got rid of a lot of the phosphorus, so if we had fertilizers that didn't use so much phosphorus, because the farmers still need to grow their crops, so it's a delicate balance.
Yeah.
What to do.
And this is why I'm so intrigued and so invested in this, because right now the state of Michigan is trying to limit the amount of phosphorus. And that largely impacts CAFO's, concentrated animal feeding operations in the lower peninsula. So right now we don't have any in the upper peninsula, but we have, I think, around 250 or so in the lower peninsula. And the question is, what types of measures, for example, when animal waste is applied to the field, what times of year it's applied, do we have to have a buffer around fields where we might have surface water runoff? And so, you know, this talk and this idea and an understanding of the real risk associated with algal blooms is so timely, because this is what's being debated right now. And so we're seeing, for example, the Farm Bureau is opposing some of these regulations, or how the regulations are being applied. So I think it's really important that we start to pay attention to phosphorus.
Very good. Well, we have 1 or 2 more minutes. Any last questions from anybody?
I have a question. Thanks for a really interesting talk. You were alluding to HABs in the Great Lakes and the air pathway. So I was wondering if you could talk about that a little bit.
Sorry, I was alluding to, I just didn't hear you properly.
When you were talking about Qatar, you were mentioning you were going to talk about the air pathway when you were getting to the Great Lakes.
Yeah, so this is something which is being explored by a colleague of ours at Cleveland Clinic in regards to Lake Erie. And what we see on the southern side of Lake Erie in the Sandusky area and other areas is that you get these hot spots of ALS. And we see this in Europe as well as in the Eastern US. If you have a water body which regularly has cyanobacterial blooms, we see hot spots of neurodegeneration on the downwind side of that lake. And so there's work coming out of Dartmouth now looking at actually cyanobacterial cells which are being inhaled, and they're finding those cells in lung tissue. And again, the toxins themselves and the cyanobacterial cells can be aerosolized. So, you know, in the Middle East, it can be a sandstorm. But here in this region, it can simply be a high wind event going over a lake, which has a cyanobacterial bloom, which can increase our exposure.
Is there any chance of it happening in Lake Superior? I mean, we live right out in the middle of a big lake, so are we safe? Or?
Yeah. Well, no, because we're starting to see more blooms in Lake Superior. So the Great Lakes, I'm sure you all saw this, was on average 11 degrees warmer this summer. And so it's the warming water temperatures, it's the increasing phosphorus, and that's what's really driving these blooms. So we can't, we can't effectively manage temperature, but we can manage phosphorus. And so that's the best way that we can address this.
So maybe our masks are good for more than just COVID, eh?
Yeah, well, yeah, that's a good way to look at it. But as Lake Superior warms, we're going to see this with increasing frequency, duration, and intensity, and we're starting to see that as well.
Well, on that very happy note, I will go ahead and wrap up the seminar. If you have time and people want to stay a little bit later to chat more, that's fine. Otherwise, it's getting to 4 o'clock. Those who have to go, that's fine too. So thanks again, Renee. I really appreciate it. And who knows, maybe some colleagues in the audience will contact you for further research. Who knows?
Yeah, no, I really enjoyed it. I think it's fantastic. Thanks for having me.
Yeah, thank you.
Thank you.
Yep, I'll hang on for a few minutes if anybody has any other additional questions.
Thank you.
Thanks.